Hello, everyone. Thanks for joining me today. My name is Stephen Burden. I'm the CEO of Facible Diagnostics. I appreciate you taking time out of your day to come listen to me talk more about myself, about my company, Facible, what we're doing, and where we're going to go in the future. Before I get started, I would like to just make a note of a disclaimer we have here. This is our Regulation A disclaimer that we're offering securities currently. We do have a offering circular that is available at the SEC at this underlined sites here and here. It's also available on our website at invest.facible.com. And so you should be fully aware of what these documents say if you're interested in investing. So now that that's out of the way, I got to do that because the lawyers tell me I have to do it. So now that that's out of the way, I'd like to just give you a little history, a little background on myself before we get going. I worked as a medical technologist in several labs. I worked in a large reference lab in Utah and Salt Lake City. I worked in small rural hospital labs. It was really at these small rural hospital labs that I saw a lot of pain. I saw a lot of things that I wanted to go change. So I was the one that was running these tests that doctors would go, okay, we need to run this test, this test, this test. I'd say, okay, here's the samples I need. This is what we're going to do. And that's really where I spent almost a decade living and thriving and really loving it. And it was really in the rural hospital systems that I saw the disparity between, people that have access to care and people that don't. And this includes testing too. And so I saw things that really made me want to go find solutions to these problems. And I saw multiple problems. And so I focused on one or two of them. And really what I saw was, people lacked access to quality diagnostics. Me being a diagnostician, this is what I'm passionate about, and this is what I saw. So I went to school to find a solution to some of these problems. And, as I worked in the labs, it became apparent to me that just sometimes the technology isn't capable of giving the results that are really needed. I ran a lot of rapid antigen tests in these small rural hospitals because that's all they really had access to, for ER situations, for clinic situations. And, I would run like a rapid strep test, and it's small, so everyone knows that I'm in the lab. All my friends that take their kids to these places, they know I'm running the test back there. And these rapid strep tests aren't very accurate. They're very fast, but they're not very accurate. And, I would get the occasional conversation with my good friends of like, oh, my kid, you said they were negative for rapid strep, but, 2 days later, we get a call from the doctor's office saying, oh, they're positive. We need to come get some antibiotics. My kid's in pain and they're suffering. Why? Why were you wrong? And first I'd say, well, it wasn't me that was wrong. This is the limitation of the test. This is the technology that is the standard of care today. And this technology has been around since the 80s and 90s. This is old technology. And they're like, well, why isn't there new stuff coming out? And I'd be like, I don't know. And so this really affected me. And I really focused on solving this problem of, fast tests aren't accurate enough and accurate tests aren't fast enough, especially at the point of care. And so there's always this trade-off between speed, accuracy, and ease of use. You saw this during the pandemic with the rapid antigen tests out there. You can take them, they're fast, you can use them yourself. It doesn't require a trained physician to use it. But if you're a negative for COVID, does it really mean you're negative? No, not necessarily. And then the flip side, at the beginning of the pandemic, we saw, PCR testing, which is very accurate, but they're not the easiest to use and they're not the fastest. And we saw turnaround times or how long you had to wait for results, up to 2 weeks. And that's really long. Normally a PCR is still a couple of days turnaround time. And so really like the state of diagnostics, even during the pandemic is still lacking for a solution that can really find that center ground between being fast, accurate, and easy to use. Dr. Fauci had a great quote at the beginning of the year. He said, we need a highly specific, highly sensitive, easy to do test at the point of care, and we're not there yet. And so even at the beginning of this year, like there still weren't any solutions that are widely available to people that they can use. Well, Facible, what we've been working on is that solution, something that finds the center of being sensitive and accurate, easy to use, and fast and portable. This is really the focus of what I did my PhD in. It's where we started Facible around. The scientists, this is what we're passionate about doing here at Facible, is using this technology that we're calling QLAD, which really does have a sensitive, accurate, easy to use, and it's fast. This is the slide that really excites me. This is talking about the technology. It's a really difficult technology to explain to everyone, but this slide really, I feel like it captures really what it does. I really enjoy this slide. And so what we have is a technology that is completely synthetic. So unlike the rapid antigen tests, we don't require animals to make our, to make our antibodies. We don't use antibodies. We use nucleic acids, and these nucleic acids are represented here in these blue T-shaped structures. And these nucleic acids form a structure. And we have here on this picture also a dye, which is DIM, represented by the gray dot here. And normally these two don't associate with each other. The dye is something that can emit light, but only under certain conditions. And so the nucleic acids in these dyes, they just kind of hang out. They don't do anything with each other. That is until a target molecule or target protein. In this case, we have a SARS-CoV-2 protein that is present by this big purple blob-looking thing right here. And when that happens, that nucleic acid structure is then free to associate or bind this fluorophore. When it does, it can emit light. And so based on the presence of light, we can know if the virus is present. And so this is a really big deal because currently in other technologies that are similar, a technology like this would require a lot of processing steps. It would require a long time to run, long incubation times, washing steps. It requires trained personnel. And even then, there's still risks of doing it wrong. And so what we did at Facible, what we really focused on was taking the complexity of that workflow and engineering it into this chemistry. So it's a very complex chemistry, but a very simple output. Is there light? If there's light, there's a virus in the sample. This can be adapted to small molecules. It can be adapted to other targets of interest, such as ions. We have a fairly wide range of flexibility as far as which target molecules we are detecting. So this chemistry really is the center of our vision here at Facible. This will be what powers our products moving forward. It's what's powered our products, up to this date. And we are just so in love with it here. We think this can handle a wide breadth of testing. And so our product, what we're really focusing on now is taking that chemistry out of the lab and making it available in a form that everybody can use it. So really what we have is this next phase that we're entering into, which is taking our chemistry and making it into a point of care system. So building this analyzer that will read the test, having a test format or workflow that anyone can use. So right now we have a 5-minute read time on our tests, so that's a very fast time. We have a high degree of accuracy right now in the chemistry, greater than 95% in its current form. We're working on this hardware piece. This will be the next development process that we're working through. And really accessible hardware is something that I should make a note of here, is that this is really important that people can access it, all without losing the sensitivity that we've demonstrated to date. And that's really important because, as I talked about the rapid antigen test earlier, they're not very sensitive. And this is a big problem when you're trying to catch infections early in the infection cycle. And really the applications for this are, you go into let's say a Walgreens or a pharmacy, you have a test taken, and while you're there picking up, Pedialyte for your kid and they're getting tested, by the time you get back, the test results are there. They're sent into your physician. This is great for rural, remote clinics, where you need really fast turnaround times. You don't have time to waste sending samples out to the next city over to get a test run. One thing that really is important for me is that we start to put more pathogens onto one test. We call this multiplexing. So being able to take multiple pathogens, test them all at the same time in the same chip, the same visit, to me really is important, because no one wants to wait for one test and then go wait for another test and then go wait for another test. Like, just doesn't work. So being able to test this all at once is really where this technology we feel is really suited to go. This is a large market. So the human health market was estimated in 2019 to be $60 billion. That's before COVID came in and blew that number out of the water. The human health market, here are some common numbers that we've seen across the globe. These are the number of cases that you see for malaria, norovirus, commonly encountered disease states that our technology is really suited to test for. For every positive case you see here, our rule of thumb is there's 4 to 10 negative tests that were run that you don't see. And so across multiple verticals such as animal health, environmental health, water testing, food testing, nutraceutical testing, and plant, crop testing, we believe this is a very large addressable market, $420 billion globally. So this is a massive market. It's ready for technology that can really access the whole market, not just a small subsector. To get there, we've been on quite the journey so far. We continue to push forward. We brought in our proof of concept technologies that we've patented. We have 3 patents pending currently right now. We're working towards our FDA EUA approval for our COVID test. But really what we've been focusing on is raising our Series A. We are using a Regulation A+ pathway. This is a equity crowdfund, or called a mini IPO, where we can raise money from accredited investors as well as retail non-accredited investors, which we're currently doing right now. This is something that really is going to drive the next phase of development for us, which is that respiratory panel, which is a 6-pathogen panel that we're really trying to get into the point of care. So bringing that chip, the analyzer, up to speed, and again, merging it with our chemistry, that's really where we're at right now. We're focusing on for our Series A funds. Following that period of time, we'll start our clinical trials, which would have to go through the FDA, most likely a 510(k) pathway, but there's still some questions there as an alternative pathways for us, which will lead us to our primary sales and long-term growth. The long-term growth data associated with this, bring it out to other panels, such as a urinary tract infection panel. These are all things that we're working on. What is really going to be the future there is something that we're working on right now. So in our milestones for this Series A, once we get the sufficient funding, we'll initiate phase 1, which we've actually already started working on. So bringing up the respiratory pathogen panel, this is flu A, likely flu A, flu B, RSV, COVID, RSV, and probably like an Epstein-Barr virus. Working on the manufacturing feasibility, the analyzer, but also the chips, running through different prototype designs for manufacturing, and really nailing down that regulatory path. We estimate that takes about 18 months to do that. In phase 2, really focusing on productization, doing beta tests with clinical partners that we've established, and working on those larger scale pilot manufacturing. In phase 3, this is the verification validation. These are our regulatory tests that we'll be running, as well as gaining that regulatory approval and hitting our initial sales. So to date, we've had a product out in the wild. We've had partners that have used it. We've done internal testing on it. And this is a test that is a moderate to high complexity test. So it still is something that has to be run in the lab. It is a version that we are using to springboard us into the next point of care adaptation. To date, it's performed really well. It has a sensitivity of 97% for an antigen test. That's great. Specificity of 100%. We have confidence intervals on these. A limit of detection of 1.88 TCID 50 per ml. And most of you probably know what that means. If we were to compare it to other antigen tests out there, that is the second most sensitive limit of detection that would be out there for antigen tests. And that puts it right in the middle range of PCR. If we're converted to PCR units. But the most important thing is that we have these stats, and it's not a long test. This has a 5-minute analyzer read time. So by the time that we put this on the analyzer, the time that it spits out results that are ready to be sent over to the physician. That's a 5-minute time. And that's a fast time right there. So you can see how this could be used in a lab to clear backlog for PCR or other antigen tests, really to get a lot of samples through in a very short amount of time. So Facible is a group of like-minded individuals that is committed to bringing hospital-grade diagnostics to everyone, regardless of their socioeconomic status. We all believe that people need access to diagnostics so they can really, truly understand and better manage their own health. We're located in Boise, Idaho. We have about 10 employees. We're founded in 2019, just before the pandemic hits. We have 6 PhDs that have been involved in this company, and we're working to expand this team as we go through our Series A and our Regulation A+. We're looking to expand this team and grow more, what we call, we need more nerds, we need more business people. We're looking to expand. And so this is something that's a really exciting phase for us, the type of people that we want to bring in and help us take us to the next level here. This is our current leadership team. See my nose itches. This is our current leadership team. I'm the CEO and the founder, followed by my co-founder, Clementine Bohachi. She's my chief science officer, another PhD here. Hunter Covert, our research and IP. He comes to us from the VA. He's a virologist. He's been one of our guys that's been here through thick and thin. He's been here since almost the beginning. We have Brent Fossen, our COO, one of the people that I brought in early because he was working in a medical device company that he started. Convinced him to jump ship and come here and help us, build this company from the ground up. Micah Cormelo, our chief of sales, has years of experience in the medical software sales side of it, so he understands the buying cycles, working with healthcare providers and big, large medical systems. Then Travis Nielsen, who's our head of marketing here, helping us share the vision to people in a way that they can understand it. Our advisory team, we have several people that have been with me for a while, guiding me through these times. Matt Hawkins runs a fintech, is also one of our lead investors. Cassie Y, a local infectious disease expert that's been guiding us in the use case. And then my boss during my PhD, Eric Hayden, who was instrumental in helping me figure out some of the science behind this and how to really get around some of those early problems that I was experiencing. We also use a group called Product LLC, the founder of which has been VP of some very large life science companies in the product side. He's been helping us with our quality systems and product and really making sure that we can make a product that has excellence, make it the same way every time, that we are doing things the right way in a very operational oriented side. And then our legal team, Perkins Coie, they head up our IP strategy. They make sure that we are protecting ourselves both intellectually and as a result financially. So some things that are interesting about this, this is more, what we see the future of this product, this 6 pathogen panel, how it's being used. These are 4 of the major, I'll say tranches, the buckets that we would expect revenue to be generated from. So we would expect something like this to be sold at $35. Our anticipated COGS are right now at $2.25. This is a little high. We think eventually at scale, these COGS can come down to, somewhere around $1. These are the 6 pathogens that we have identified initially that we would like to launch with. And you can see here some of these numbers, the penetration rates, the number of hospitals in the US, the urgent care clinics, long-term care facilities, and some of the revenue that we would expect just from these 4 buckets alone. You can see that, if you can really nail the technology, that this is a technology that we think people will want in their systems. So on the exit side, there's 2 paths that we have identified that really do fit us. One, because we've already started this IPO, and IPO is actually something that we could consider in the future. You can see some of the other comparables here that have done an IPO and what their market caps are. Market cap, or the IPOs in this space, they can be big ones. They can be really big. And so we think, having control of the company, bringing it to an IPO, that's something that really fits our vision. But however, more likely is something like an M&A. where a merger and acquisition, where a bigger group that does really good at distribution, they have their sales channels already built up. You can see here some examples of mergers and acquisitions, Mesa Biotech, Luminex, GeneMark. All of these were very successful acquisitions by their parent companies. So really, these are the two exit strategies that we have in place right now that we're looking at. And both of these are very good opportunities for us. So the raise, like I said, we're using this Regulation A+. It's termed the mini IPO. We can sell to retail investors our private securities, much like we would to accredited investors. And to do this, we have a firm of teams that have come in to help us run this raise. We have a transfer agent that has come in that manages our cap table, make sure everyone's in compliance. We have a broker dealer here that comes in and has all the licensures and makes sure that we follow the Know Your Investor laws and the anti-money laundering laws. We have an investor acquisition group that goes out there and advertises these securities to people, both accredited, non-accredited. And we have a market intelligence team that is coming in saying, hey, this is how much you should expect to raise, this is what the price should be. And so all these things come together. We decided to go after a raise of $40 million. We don't have to raise a full 40. We could stop early if we feel like that is a suitable situation for us. We're raising at $5.80 a share. This is currently what we're undergoing right now. And the use of these funds will be to take our technology out of the lab and bring it to the point of care over-the-counter format using this, and it will be done for these 6 respiratory pathogens. So that is the slide. I'd like to just open up the time for questions. I saw that we have one come in right now. I appreciate the patience here. So if you have any questions, please go ahead and type them into the questions box. I'll read them and I'll answer them. So this one is, how protected is your IP through patents, both methodology and device applications? What time horizon have you set, quote, goal to sell off this technology to a bidder to allow investors to profit? That is a good question. So this is a technology, if you remember, that I worked on in my PhD. And so this isn't a technology that exists on the market. This is something that we are translating out of research and academia into development. And as such, the patent space for this technology is fairly open. So as we worked with our IP lawyers, we were guided, and they have told us that this is a very broad patent because the IP space is fairly open. And this allows us to be able to carve out, smaller patents, more narrow patents in the future. So this is a family of patents that we're looking at. Much like if you were to be on the early end of PCR, what those patents look like, instead of just having a better instrument or a better widget that uses it, this is a chemistry patent that we think can be very broad and very defendable. So what is the time horizon? I think the time horizon is set in the exit strategy for the M&A. That would be, those people are interested in the technology and acquiring the technology. And that's, 3 to 4 years. We feel that's where we'll be best suited with an analyzer, with the chemistry, and with an assay. So I hope that will answer your questions there. So next question. How have or will current market conditions, public markets are down, inflation is high, affect Facible? That's a great question. We've had that question, and I've been asking, and I've been listening. I've had my ear to the ground on this one. I've been asking our brokers, our transfer agent, our intelligence group, our investor acquisition groups, because they're all involved in other areas of this Reg A as well. So they have a lot of data flowing into them. They're digesting it, and they're passing it on to me. And what they're passing on to me is that this market, the Regulation A+ market, the retail offering in these private securities is strong. We are seeing a little bit of uptick in investor acquisition costs to acquire it, but we are still seeing people come into investments. They're seeing a strong sentiment towards these. These aren't affected by market volatility like the public markets are. And so really people are kind of looking at these as more of a moderate term play. They're putting their investments in it for a year to 3 years versus trying to get something out of it next month or next tomorrow. And so they're really concerned about the public fluctuations, and we don't have those over here because our stocks aren't being publicly traded. And so really that's what I've heard, and that's what we're seeing. People are still coming in. They're coming into Facible, they're coming into other people, and people are really excited to invest in a company before it gets acquired by a Roche or BD. These pre-IPO companies, people are really excited to get in on the ground, get on the ground floor and invest in these companies. So third question, how do you differentiate from other competitors? That really is based on our chemistry. Like I said, this chemistry is new. It's exciting. It hasn't hit the market in a broad way. It's not PCR, which is a bulk of molecular testing. Is a bulk of what the COVID testing was in the labs was molecular tests. It's not a rapid antigen test or anything close to it. Which those are the two kind of buckets that people try and group us in. Are you a rapid antigen test or are you a molecular PCR test? Yeah, we're neither. We are something new. We are something that is flexible, something that is fast without sacrificing the accuracy that you would get in PCR. And so really, that is the differentiator. It's our chemistry. It's the center of our platform. It's the center of our future ecosystem. And to me, in my personal humble opinion here, this is the future of rapid diagnostics. This is what I am building the company for. This is the vision. This is what we're passionate about, and that's what we're building it on, is that chemistry. Can you elaborate on how Facible will use the money that they're raising to further develop your product and to bring it to market? Yeah, I can. Right now, we got the chemistry in a good spot. It's in the lab. It kind of negates some of the value proposition of speed if it stays in the lab. The money that we are raising right now will go towards analyzer development. It'll go towards assay development. It'll go towards continuation of our chemistry development. And it'll go towards the merging of all three of these components. It'll go towards bringing in or doing our clinical studies. It'll go towards working on our current chemistry to bring it into the EUA, which is the emergency use authorization by the FDA. That is what the money will be spent on. We'll have to, work on partnerships to bring in a hardware partner, which we've identified. And so all those things are going to go towards those three components: the assay, the chemistry, and the hardware. So can you explain more of the BOGO shares and when it ends? So right now, to kind of get some momentum early in our raise, we've established a buy one, get one for our securities, so for our stock. So right now, if people invest in our stock at $5.80 a share, they will get additional stock on top of it. So buy one, get one. This actually has been going on for almost 3 weeks now, and it'll be ending actually tomorrow. So if you know anyone that wants to invest and they want to get a screaming deal, buy one, get one, I would recommend that you tell them now and you talk to them about it, because it's going to end tomorrow. So what is the stability of the reagents to run compared to PCR primer probes? Oh, I know the person asked this question. That's a good question. It's a very technical question. Right now, we don't have any proteins in our assay. And so what the whole goal is that these are cold chain free. These are room temperature reagents. And these are something that we can use in a wide variety of temperatures. So think Africa versus Norway. So right now, we have good shelf stability. We have room temperature running. So in the lab, controlled environment. The one thing that we have temperature control on is the actual sample itself. So if the sample needs to be shipped cold or frozen, just to preserve the actual proteins in the sample. Our reagents, as of now, and I don't see this changing in the future, is room temperature, storage room temperature, run at room temperature. I don't want any cold chain involved in our production or storage in our shipping. What is the development time for a new detection target? So we did our original COVID probe that is currently on the market right now. We did that about 3 weeks. It took us a lot longer to get it into the assay itself, but to develop the probes, we're trying to hit this 3 to 6 week turnaround time. And that's really been the focus of what we've been doing the last 2 to 3 months, is making that process to get it consistently 3 to 4 weeks, consistently putting processes in place in our pipeline to get that the same every time. So we don't have to worry about, well, is this one going to take us 5 times as long? Well, if we have the right processes in place, the right procedures in place, we believe that this is a scalable process development pipeline that we can turn new probes around in 3 to 4 weeks. We're not quite there yet. We're a little bit longer just because we've been doing some basic research on that pipeline. But I believe we are almost done with this process of making it scalable and repeatable and reproducible. We should be almost there. And not only that, I think that we'll be able to do multiples of these in parallel. So that's something that, because ideally we want to have, we're saying the 6-plex pathogen panel, that's how many pathogens we'll be testing for, but we'll probably have multiple probes in there. And if we want to break out to 30 plus, like I'm driving for in the future, having 30 plus pathogens on a single test, that would be something that we'll have to, scale the development pipeline for. And so this has been the focus for us for the last 2 to 3 years. Good question. All right. So, that's all the questions I have so far. We'll wait a minute, see if anybody has any other new questions in there. But I appreciate everyone's time. If you have any questions for me, my email is available. We will send out a copy of this deck to everyone on our list today, so you have a copy of it. And our contact information is on there. So if that is all said and done, if there's no more questions, or if you do have questions, please email me. I think that's wrap it up, and we'll let you guys go. I appreciate your time. Thank you.